This method can help answer key questions in tissue mechanics, such as how properties of cell-cell contacts change in space and time during tissue morphogenesis. The main advantage of this technique is that it is weakly invasive and is compatible with live imaging such as light sheet microscopy.
This technique doesn't require injection of beads into the tissue, usually used as intermediate probes on which optical forces are exerted.
Demonstrating the procedure will be Claire Chardès, an engineer, and Raphael Clement, a research scientist from my laboratory.
To begin, set up an upright microscope for light sheet microscopy in the horizontal plane. Then, prepare the laser and optics to act as the optical tweezers. Next, pipette 1 microliter of 500 nanometer fluorescent beads into a glass cuvette and add 10 milliliters of distilled water. Place the cuvette in the cuvette holder under the objective and adjust the focus of the objective so that the objective touches the water's surface. Now, open the Integrated Development Environment (Qt Creator), which is used to control the acquisition software. Open the acquisition software by opening the microrio.pro file and turn on the live acquisition mode by selecting Live. Prior to switching on the laser, put on safety goggles. Then, adjust the laser power to 1 watt and switch on the laser. The live feed should now show a bead trapped at the laser's focus. If necessary, adjust the position of the second telescope lens to observe the bead in focus. One may also need to adjust the angle of the periscope mirrors to center the bead in the image.